Have you ever thought why some odors are liked by someone but disliked by someone else? And why your eye color is black but other person may have blue eyes? So, why we are different from each other? Well, this difference is due to the difference in genetic material of the organisms. A genome is an organism's complete set of genetic instructions. Each genome contains all of the information needed to build that organism and allow it to grow and develop. Let's understand it. Our bodies are made up of millions of cells, each with their own complete set of instructions for making us like a recipe book for the body. This set of instructions is known as our genome and is made up of deoxyribonucleic acid. Each cell in the body, for example, a skin cell or a liver cell, contains this same set of instructions. Well, genes are small sections of deoxyribonucleic acid, or DNA, within the genome. These genes contain the instruction of biological molecules, usually proteins. They contain the instructions for our individual characteristics, like hair color, such as brown, or black hair color. Well, genes also control different characteristics, such as eye color, green eye color, or blue eye color, and height, short or tall. But genes encodes only 2% of our all genetic information. The human genome is made of 3.2 billion bases of DNA, but other organisms have different genome sizes. Your genome is made of a chemical called deoxyribonucleic acid, or DNA for short. So, DNA, or deoxyribonucleic acid, is a long molecule that contains our unique genetic code, like a recipe book. It holds the instructions for making all the proteins in our bodies. All living things have a unique deoxyribonucleic acid, or DNA, just like every person has its own identity card. The order or sequence of these bases form the instructions in the genome. Deoxyribonucleic acid is a two-stranded molecule. Deoxyribonucleic acid has a unique double helix shape. Like a twisted ladder, deoxyribonucleic acid contains 4 basic building blocks, or bases. These are called as adenine, cytosine, guanine, and thymine. So each strand is composed of long sequences of the 4 bases: adenine, cytosine, guanine, and thymine. The bases on one strand of the deoxyribonucleic acid molecule pair together with complementary bases on the opposite strand of deoxyribonucleic acid to form the rungs of the deoxyribonucleic acid, or ladder. The bases always pair together in the same way: adenine with thymine and cytosine with guanine. Each base pair is joined together by hydrogen bonds. So, adenine and thymine has 2 bonds, but cytosine and guanine has 3 bonds. You can remember these pairs using a mnemonic: apples in the tree, and car in the garage. A for adenine, T for thymine, C for cytosine, and G for guanine. Each strand of deoxyribonucleic acid has a beginning and an end called 5' and 3' respectively. The two strands run in the opposite direction, called as antiparallel to each other, so that one runs 5' to 3', and one runs 3' to 5'. They are called the sense strand and the antisense strand, respectively. The strands are separated during deoxyribonucleic acid replication. This double helix structure was first discovered by Francis Crick and James Watson, with the help of Rosalind Franklin and Maurice Wilkins. The human genome is made of 3.2 billion bases of deoxyribonucleic acid. But other organisms have different genome sizes. Chromosomes are bundles of tightly coiled deoxyribonucleic acid, located within the nucleus of almost every cell in our body. Humans have 23 pairs of chromosomes. 22 pairs are autosomal chromosomes, means non-sex chromosome, but one pair contains sex chromosomes. In plant and animal cells, deoxyribonucleic acid is tightly packaged into thread-like structures, called chromosomes. This is in contrast to bacteria, where deoxyribonucleic acid floats freely around the cell. A single length of deoxyribonucleic acid is wrapped many times around lots of proteins, called histones, to form structures called nucleosomes. These nucleosomes then coil up tightly to create chromatin loops. The chromatin loops are then wrapped around each other to make a full chromosome. Each chromosome has two short arms, called as p-arms, two longer arms, called as q-arms, and a centromere, holding it all together at the center. At the ends of each of our chromosomes are sections of deoxyribonucleic acid called telomeres. Telomeres protect the ends of the chromosomes during deoxyribonucleic acid replication by forming a cap, much like the plastic tip on a shoelace. Humans have 23 pairs of chromosomes, 46 in total. One set comes from your mother, and one set comes from your father. Of these 23 pairs, 22 pairs are autosomes, means non-sex chromosomes, and look the same for both males and females. One pair are sex chromosomes, so differ depending on whether you are male or female. If X chromosome comes from mother and Y chromosome comes from father, gender of the baby will be male. If X chromosome comes from mother and X chromosome comes from father, gender of the baby will be female. So we can say that genes are small sections of deoxyribonucleic acid within the genome, and the deoxyribonucleic acid making up each of our chromosomes. Gene therapy is the manipulation of genes, adding deoxyribonucleic acid to the genome, or altering the deoxyribonucleic acid of a gene in order to cure a disease, if doctors can access the tissue easily, but the cells cannot be removed from the patient's body. The technique of choice is in vivo gene therapy, in which genes are delivered directly to somatic cells. The therapeutic gene could be injected into retinal cells, for example, or inhaled into the lungs. These types of delivery methods are easy to perform, but they can introduce the deoxyribonucleic acid into only a fraction of the affected cells. For other diseases in which the affected cells can be removed from the body, more potent ex vivo gene therapy can be used. Here, researchers remove tissues, such as blood precursor cells from the patient's bone marrow, expose the cells to large amounts of the therapy gene, while the cells grow briefly in culture, and then return the cells to the patient's body for either in vivo or ex vivo approaches. The foreign deoxyribonucleic acid needs to be packaged in some kind of vector, so that it can be taken up by enough cells for successful therapy. Most gene therapy trials have been performed with either of two types of viral vectors: retroviral vectors or adeno-associated viral vectors. In both cases, specialized cell lines package the therapeutic deoxyribonucleic acid into virus-like particles. Let's take example of both in vivo gene therapy and ex vivo gene therapy in case of in vivo, in which cells cannot be removed from the patient's body. In indicates for inside the body. Modified deoxyribonucleic acid injected into vector. Vector binds to cell membrane. Vector is packed in vesicle. Vesicle breaks down and release vector. Vector then injects new gene into nucleus. Cell then makes protein using new genes. In case of ex vivo gene therapy, cells are harvested from patient, in which the affected cells can be removed from the body, as X means outside the body. Cells are harvested from patient, virus altered, so cannot reproduce. A gene is inserted into virus. Altered virus mixed with cells of patient. Cells become transgenic. Altered cells are injected into the body of patient, and the finally altered cells produced desired proteins. So, this was all about today's video. Stay tuned to sqadia.com for more precise and knowledge-stuffed videos.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.